Hello, colleagues. I am pleased to have the opportunity to present the Advanced Biopharmaceuticals Product Approval Education Workshop. I will be presenting the Chemistry, Manufacturing, and Controls, or CMC session. My name is Holly McCaleb, and I am a director with the Pfizer Global Regulatory Sciences CMC organization. I have been with this department for over 9 years and have spent the last 6 years focusing on gene therapies. Today, I will be speaking to you about unique quality and regulatory considerations for AAV gene therapies. I will start with unique challenges to AAV gene therapies overall, then move to drug substance manufacturing topics, drug product and supply chain considerations, analytical topics, regulatory considerations, and finally, a review of discussions with FDA and EMA during clinical development. I would like to note that in some cases there is an R in front of the AAV to denote reference to recombinant adeno-associated virus products. For the purposes of this presentation, RAAV or AAV designations will be used interchangeably. We will start with unique challenges to AAV gene therapeutics. The AAV vector consists of two parts: the AAV capsid and the DNA with the gene of interest, which is also known as the vector genome. The AAV capsid is composed of 60 protein monomers in a ratio of approximately 1:1:8 of VP1 to VP2 to VP3. 5 AAV proteins are required for replication and assembly. 4 adenovirus encoding genes and 1 non-coding RNA gene are required for expression and assembly. The DNA with the gene of interest is encapsulated within the AAV capsid. Because of their complex nature, AAV products are significantly more challenging to make compared to recombinant proteins. While there are multiple ways to manufacture AAV capsids, my slides will focus on the manufacturing processes used at Pfizer. The AAV manufacturing process starts with the cell bank, usually HEK293 or HEK293 cells, and the 3 plasmids necessary to generate the AAV. Upstream processing includes growth of the cells in culture, transient transfection of the HEK293 cells with the plasmids, and then harvest of the AAV capsids. Downstream processing includes purification, viral clearance steps, and concentration. Filter, fill, and finish steps are completed to generate the drug product, which is then stored at temperatures below -60 degrees Celsius. An electron microscopy image of purified AAV capsids is shown on the bottom left of the screen. AAV dose requirements can vary widely depending on the indication and the target tissue. These differences can mean AAV gene therapies must be manufactured to accommodate dose requirements that can range over 4 orders of magnitude. The images of the planets on this slide demonstrate those relative dose differences. With dose requirements spanning this large range, not all AAV products will have the same yield requirements. Key challenges for AAV manufacturing are summed up on this slide: low productivity, low concentration, low availability, low volumes, limited time, and limited understanding. All of these complexities must be considered when developing a gene therapy. Dosing patients with gene therapy is its own unique journey. Patients are screened to determine if they qualify for the clinical trial. The patient is then randomized and enrolled into the study, and the patient-specific drug supply is shipped to the site in time for patient dosing. This is also called on-demand clinical supply, or ODCS. The patient is dosed and then monitored for a specific period of time. This pathway is different from CAR T therapies, where patient enrollment triggers manufacturing, not shipping. I will now move on to talk specifically about drug substance manufacturing considerations. I mentioned earlier that AAV production is generated through transient transfection of the HEK cells. This transfection uses 3 plasmids, which are the transgene plasmid, the helper plasmid, and the RepCap plasmid. The transgene plasmid includes the promoter, gene of interest, poly-A tail, and has inverted terminal repeats, or ITRs, on the 5' and 3' ends. The helper plasmid contains adenovirus elements needed for replication and packaging of the vector genome. The RepCap plasmid contains the genes that code for the replication machinery in capsid proteins and defines the tissue tropism. For example, AAV2 capsids would be targeted to the kidney. All 3 plasmids must be present during transfection to generate the AAV product. Key considerations for drug substance manufacturing include, first, that drug substance manufacture is time-consuming, complex, and expensive. Second, teams are often faced with material limitations due to low titers, low yields, and a relatively high percentage of the batch going to GMP testing. Third, process understanding is still evolving. We know the separation of empty particles, which are capsids that do not contain the gene of interest, and full particles can be challenging, and viral clearance must be demonstrated as well. Manufacturers must plan carefully to execute the necessary development work to support the registration package. Now I will talk about drug product and supply chain considerations. Gene therapies face formulation, process, and delivery challenges. Formulation development can be difficult due to the scarcity of material. Adsorption of capsids to product contact surfaces during manufacturing and dosing must be considered. The drug product process also has to be designed to handle small volumes, and the impact of different product contact materials must be considered. Delivery of the product to the patient can require different dose calculations for actual or nominal dosing. As mentioned earlier, a frozen supply chain is usually required, and dosing is typically accompanied with an additional component, such as human serum albumin, or HSA. Many other challenges are listed on this slide as well, and all need to be considered when developing a gene therapy drug product presentation. Key considerations for drug product and supply chain activities include, first, options for material to be held as drug substance or to proceed forward directly into drug product. Second, a possible lack of drug substance stability data if the material is not held as drug substance. Third, challenges with shipping and storage of vials below -60 degrees Celsius. Flexibility is needed with label content requirements due to the vial size, and expiry is controlled through inventory systems rather than primary labels to allow extensions without thawing the vials to relabel. On-demand supply and frozen shipping temperatures require unique supply chain considerations compared to other types of biologics. Now I will talk about analytical considerations. Multiple quality attributes are evaluated as part of a robust analytical control strategy. This control strategy can include routine product testing, as well as stage-appropriate qualification and testing of process-related impurities. Many of these tests probably look familiar, as they are used to evaluate other biologic products, but some are unique to AAV gene therapies. I will discuss those further on the next slide. Key areas for AAV analytics include titer, potency, and purity. Titer is the concentration of viral particles containing the gene of interest. Multiple types of assays can be used to measure viral genome or vector genome, or VG titer, but the assay needs to be product specific, as it is critical to correlate dose throughout product development. Many early studies start off dosing with actual lot titer, then transition to nominal dosing for late stage or commercial. Potency assays are needed to demonstrate infectivity, expression, and function of the AAV. A single assay may be acceptable for early clinical studies, but multiple assays could be necessary for pivotal studies. Finally, purity is an important aspect of AAV quality and includes control of product variants, measurements of full and empty capsids, and monitoring of residuals. Key analytical considerations include, first, aggressive timelines are often needed to meet clinical development expectations. Second, attribute criticality is still an early understanding and could have high uncertainty due to limited material and high variability with assays and processes. Third, limited material can also make it challenging to develop and validate assays. And finally, most process changes will need analytical support in the form of analytical comparability. It is desirable to have changes implemented before the pivotal study, but this is not always possible. Depending on the stage of development, the analytical comparability assessments can be complex, and a statistical analysis of the comparability data is usually required to confirm analytical comparability between pre-change and post-change material. I will now discuss regulatory considerations. CMC regulatory guidance for cell and gene therapies does exist in the US and EU, but it continues to evolve with increased product understanding and the development of new technologies. One example is a recent FDA draft guidance on gene editing technology that was released in 2022. ICH quality documents are considered relevant to qualifying gene therapy products. Regulators also seem to understand the need for flexibility. Some examples of this flexibility include development of GMP guidance that's specific to advanced therapy medicinal products, or ATMPs, in Europe, expedited approval pathways, and a willingness to minimize local batch testing to reserve material for patients. Key regulatory considerations include: first, gene therapies go through standard development timelines, although they are often accelerated. Second, there are guidance documents to address issues unique to gene therapies, such as potential variability in the final product, real-time release and alternative sampling strategies to take material limitations into account, concurrent process validation, and provisions for release of material that is out of specification, or OOS. Third, GMP principles apply to gene therapies, including the cell banks, plasmid manufacturing, and AAV manufacturing. And finally, there is an expectation that the control strategy should be in place at the time of registration and should be based on the principles of quality risk management. Regulatory harmonization is crucial to development of gene therapies, as our ability to tailor CMC requirements will be limited. Lack of global regulatory harmonization may lead to increased manufacturing costs, barriers to innovation and continuous improvement, overcomplicated supply chains, increased quality risks, and reduced quality assurance. Consequences of these can be delays in getting therapies to patients, drug shortages, increased burden for regulators, and reduced trust in supply chains. Within gene therapy, it will be critical to achieve global agreement for CMC aspects. I will conclude my presentation with a discussion of our interactions with FDA and EMA. As I mentioned earlier, US and EU guidance for gene therapies continues to evolve. Both the FDA and EMA offer opportunities for increased dialogue during clinical development, including RMAT, or Regenerative Medicine Advanced Therapies, for the FDA, and PRIME designation for EMA. Some of the key CMC topics discussed during development include BG titer method qualification and validation, process changes and analytical comparability exercises, potency method development, manufacture and control of plasmid raw materials, and drug substance and drug product specifications. Manufacturers are encouraged to take advantage of the opportunities for dialogue to discuss these topics. In summary, I would like to reiterate that AAV gene therapies are more difficult to manufacture than traditional biologics. AAV manufacturing processes deliver less material than biologics, so fewer doses are made. These dose levels for gene therapies can vary significantly depending on the indication and the target tissue. Most gene therapy products require an on-demand supply chain that can maintain frozen storage, and analytical methods and quality attribute understanding is still evolving. Opportunities for discussions with regulatory agencies during clinical development can ensure industry and regulators are aligned on expectations for product quality. Thank you for your time. I do have some questions that were submitted that I can read and provide responses for. The first is, what are the criteria for full and empty capsid ratios? Empty capsids are recognized by the FDA as an impurity. They have been noted to have a possible impact on product safety. For this reason, the FDA requires measurements of percent full and percent empty capsids as part of lot release. The FDA has not mandated a particular ratio or level of percent empty particles, as the impact of their presence can vary depending on the indication and the dose level. Companies are encouraged to monitor full and empty ratios for clinical lots to evaluate consistency in the levels of the empty capsids in those final drug product lots and determine an appropriate commercial specification. Learnings on empty and full capsid ratios are ongoing. A second question submitted is, as a method of vector genome measurement, should both qPCR and ddPCR analysis results be used? If the results from the two different measurements don't match, which results should we trust? qPCR and ddPCR are both used as measurements for vector genome titer. Neither measurement is considered better or worse than the other, and it's not surprising that the results may not match because the technologies used to calculate the titer are different between the two assays. Companies instead are encouraged to try and use the same method throughout development so that titer values can be compared across clinical lots. Any switch in the vector titer methodology will require extensive bridging to understand any differences in reported results between the methods. A third question submitted is, what is the recommended empty capsid ratio before conducting the first in-human clinical trial? There is no specific ratio for full or empty capsids that is required to start a first-in-human clinical trial. Manufacturing processes should be developed to reduce levels of empty capsids as much as possible, but no specific ratio is required since the impact of empty capsids can vary depending on the indication and the product dose.